CCBR organizers, and thanks to Professor Mruganka for giving me a chance to collaborate and present work here today. I'll give a bit of a background to this work. I mean, some of this work we have been doing before we came into touch with CCBR. So what I'm going to talk about is modeling neuron-astrocyte-vessel interactions. As we all know, I mean, neurons and like any other cell in the body get their energy sources from blood vessels. Only thing is this mediation between capillaries or blood vessels and neurons happens through a mediator called an astrocyte. So basically vessels release glucose and oxygen, which are the resources for energy, which are picked up by the astrocyte. It converts it into lactate, and this is one possible pathway. And an astrocyte releases these substrates to vessel to the neuron, and neuron converts it into ATP and uses it for energy. So this, like any other cell in the body, it uses it draws its energy from the blood vessels. So the same thing is summarized here. So neurons release a neurotransmitter like glutamate, for example, which acts on the astrocyte, activates astrocyte. Lots of things happen here. Then that releases vasodilators, which will dilate the vessel. Vessels release glucose and picked up by astrocyte, converted to lactate and ATP. Okay, so the reason actually we got into astrocyte, whole astrocyte business is very interesting. We were earlier working on modeling vasomotion. So vessels have spontaneous oscillation called vasomotion, and it's there in pretty much every tissue which is perfused by blood vessels. So we were doing some work on modeling vasomotion in skeletal muscle. And if you look at the simple section of a skeletal muscle, they have these motor fibers which are surrounded by a bunch of microvessels. So here is a kind of idealized picture of that. So if vessels are oscillating, then what kind of an oscillations of the combined oscillations of all the vessels surrounding a motor fiber that are ideal for optimal delivery of energy to the motor unit. So this was the question we had in the mind. And so then I thought like this is very interesting because our conclusion is that you need to have desynchronous oscillations of the vessels for optimal delivery of energy to the motor fiber. That's very interesting because then if you apply these ideas to brain, what happens? So how do neurons and vessels talk to each other? So what kind of dynamical conditions are necessary for neurons and vessels to talk to each other in optimal fashion? Yeah.
Are you saying that the vasomotion is necessary for better?
No, it is there. Now that it is there, what are the ideal conditions of the rhythm? Okay, so so we modeled every vessel as an oscillator. So if that is the case, then how do you apply these ideas to the brain? So that's how I got into. So then I realized that unlike other tissues where the metabolizing tissue and vessels talk to each other directly, right? You say I'm hungry and send out a bunch of molecules, and then vessels dilate and release energy. But that doesn't seem to be the case with in the brain because there is this mediating glial layer. So that's very interesting. And then the question is, why do you need these glial cells? Why are they in the first place? And how come other tissues don't have? Why are there glial cells in the brain? So that gave rise to our first model. It's a very slightly abstract model, but kind of sets the stage for more subsequent work that we have done in this area. It was done as an undergrad project. So the logic here goes like this. It is like a typical engineering approach to the problem. So you have say a neuron here and a blood vessel here. Neuron is hungry and releases some sort of substrates, some sort of dilators, and vessel dilates and releases energy. So if you had that kind of one-to-one relationship between neurons and vessels, life would have been very simple. But that's not the case. You have a whole bunch of neurons and a whole bunch of vessels, right? And when neuron sends out a signal, it is picked up by a whole bunch of vessels. When vessels release the glucose and these other energy substrates, it goes to a whole lot of neurons, right? And then so it's a many-to-many connection between neurons and blood vessels. Now, how do you so now if you design this like an engineer, one condition you would like to satisfy is like this. So this neuron is like size represents its activity levels. This neuron is working very hard, spiking away at high rate. This neuron is kind of not very active. So which means the total energy influx into this neuron should be high, total energy into this should be low. Okay, now how do you arrange for this result, this general condition, by somehow tweaking the connections between neurons and blood vessels? Okay, so the thing is there should be a precisely patterned match between neural demand as defined by the spiking rates and all that, and energy supply. Okay, so how do you do that? So that means your problem is posed like this. You have a layer of neurons, then talking to a glia, specifically astrocytes, then blood vessels, and signal goes all the way here, vessels dilate, right, in a patterned fashion, and then feedback to astrocytes, then again all this is mixed and matched, and feedback to neurons. So basically this pattern, this vector and this vector should match. Now, if you forget for a moment that these are neurons and these are glia, these are blood vessels, if you think of it as just three networks talking to each other with a kind of a matching condition here, this is basically a deep network. This is basically what's called a deep belief network, right? I mean, if you take a network like this, just think of all of them as neurons, right? If you want to design a network with, if you take a deep belief network proposed by Hinton et al. In 2006, right, an input layer where you present a pattern, the last layer recognizes the pattern, right, and the feedback connections, right, reconstruct the pattern. So therefore, the feed-forward pass is called recognition, and feedback pass is called generation. So you reconstruct the input pattern, right, by the feedback. Now, this is exactly kind of architecture, that can be superimposed onto this, biological system consisting of neurons, glial cells, astrocytes, and blood vessels. So we just did a very toy model. I mean, kind of, hardcore biologists will jump at me if you say that this is a model of neuron, astrocyte, and blood vessel. But I think as a cartoon model, it's okay. So we just took two networks, two architectures. In one, it's hypothetical architecture where neurons are directly talking to vessels, and another you have an intermediate layer of glial cells. Now I trained this on a bunch of simple patterns like digits. Okay, so the idea is, so these are like patterns which we use for training. The idea, and this is the feedback that is coming from the glial layer, which is like the energy image that the neural network is receiving. Ideally, these two should look alike. Okay, so but if I don't have the intermediate layer of glial cells, they don't look alike at all. There's no resemblance. Now we add a glial layer, but gradually increase the size of the glial layer. So 10 by 10 still is pretty bad, and so then we have 15 by 15 still pretty bad. 20 by 20 suddenly things start getting interesting. Okay, the feedback energy image, right, looks a lot like the input this thing. So this is a kind of matching condition we are trying to get. Now remember that this matching condition will be satisfied only for this particular set of patterns, because the network is trained for these patterns. If you add more, then you'll have degradation in this matching. You'll have to train further, or maybe increase the glial layer size further. Because for these 6 patterns, for this critical size of about 30 by 30 size of glial layer, it worked, and there was some level of matching. So it's a very simple model, but it's a very interesting model conceptually, because you can draw a lot of, it will indicate a lot of very interesting biological consequences. So let us go over that. First of all, there are 2 directions in which signal flows in this network. There is feed-forward influence from neuron to astrocyte. Now here we are talking about something like a weight. Normally weight is something associated with the synapse, but here the idea is extended to neuron astrocyte connections. Okay, and you can give some biological interpretation to that. And on top of that, astrocyte vessel connection, that's even more interesting. So can we give a biological interpretation to that? We can. So maybe that is basically what it is telling is, for a given level of vasodilator released by the astrocyte, how much does the target vessel dilate? Okay, I can put a number on that, that relationship. Right now, I'm saying that's plastic. So in fact, since it's just a deep belief network, all the training is happening by some sort of a variation of Hebbian learning. So what we are saying basically is, neuron-astrocyte connections are, satisfy some kind of, No, this is a very abstract, simplified network. And we'll get it. I'll show you a more biophysical model after this. It's just a starter, because it's a very big picture, and we are covering all 3 in one picture. And just to get 2 points, I'll come to that. So the reverse influence is astrocyte to vessel. There also we are saying there's some sort of connection strength, and that's plastic. And from astrocyte to neuron is also plastic. Those connections are also plastic. Okay, so now, is there plasticity in these networks? I mean, or is it just wild imagination of a modeler? So in the model, connections are trained by Hebb's rule, and okay, so activity-dependent neuron glial remodeling in adult hypothalamus. Okay, this study. Notion of metabolic plasticity. So mesostudy is a big name in the whole field of energetics and astrocyte energetics and all that. And he talks about the notion of metabolic plasticity. It is as though brain is tuned to deliver energy in an optimal way for certain ongoing activity patterns. If you change the activity pattern, then your delivery may not be optimal. Okay, so the plasticity is there, the adaptivity is there, so that the network gets tuned to support certain kinds of activity which are generally found in the brain. But the mesostudy doesn't give a kind of a quantitative or even a cellular level mechanism for the plasticity. It's just based on experimental studies, he says that there should be some kind of activity-dependent plasticity going on. So we have a model which actually gives you kind of a concrete mechanism for that. Now, one more thing that's happening here is, in the model, we were able to achieve this matching. So key thing is this matching condition. Once you assume that matching principle is a valid principle to think about neuron-astrocyte-vessel system, we have a whole lot of consequences. I just showed you the consequences regarding plasticity. Now, what about neuron-astrocyte ratio? So one thing we find here is that to support these 6 patterns and get this matching condition, you need to have a sufficiently large glial layer. Now, glial layer, I mean, we know that the number of astrocytes is much more than the number of neurons, something like even double. People talked about 50 and 10 and all that, but 2 seems to be like a reasonable figure that's floated around these days. So in the model also we have this kind of a relationship. And local density of glial cells is an indicator of metabolic demand of local neural tissue. One more thing is very interesting, is glial neuron ratio increases with higher environmental enrichment and stimulation. So if you grow an animal in a rich, sensory rich environment, its glial neuron ratios go up. That's exactly what we are seeing here. So to support certain kinds of complex pattern activity in the brain, you need to have sufficiently large number of astrocytes in that area, whichever area of the brain is active. So now, let's review some of the conclusions. So brain's computations are performed by the coordinate function of neurons, astrocytes, and blood vessels. So now can we talk about vascular computation? That means, so basically mathematically, I'm looking at 3 networks here. And in fact, in terms of training this triple system, I'm using learning rules, which we've always been using only for neurons, right? If that is the case, where do I draw the line? Why do I say, and it's just being completely prejudiced to say that neurons alone do all the computation, they're the computational heroes of the brain, whereas, the sort of astrocytes do all what we call, they're sidekicks, right? They're doing sidey jobs, and vessels are just like this kind of passive irrigation system. And this kind of we have to question this kind of viewpoint. Now, do arterioles have tuned responses? So that is, if I show an oriented bar, into the visual cortex, then are likely to find an arteriole which will respond and dilate specifically in response to that kind of oriented bar. Very likely, I would bet on it, that's going to happen. In fact, that's what you're actually seeing in your functional imaging. Only thing is you don't think of it on those lines. Right? So then what about other analogs of neural computational features in cerebral vasculature? So people are, okay, I'll come to that in a second. So are there topographic maps in the microvascular bed? Can you find orientation maps? Can you find direction sensitivity maps and color maps and so on and so forth? Will you find attractors? Because vessels are dynamic. We think of them as a dumb, bunch of pipes, but they are very dynamic. There's oscillations, there's rich dynamics, right? People have studied that extensively. In the vascular biology guys do study this. What about synchronization, feature binding? Okay, but the oscillations are much slower than neural oscillations, but their oscillations are all the same. Right? What about plasticity? We have just talked about that. So that will open up a huge, it's a total paradigm shift, I would like to say. And with this, the number of experiments you have to do to confirm different parts of this basically conceptual framework is quite high, and it's quite exciting to think about that. So when I talked about this, people said this is a very abstract model. What is this? You take a deep belief network and just randomly superimpose it on a very different biological system, and then you tell story after story. Yeah. So come up with something real. Be real, right? So then we did a very detailed model of a single neuron and single astrocyte and a single vessel, right? And lots of biophysics. So for example, astrocyte you have IP3 activation, then calcium release from the internal stores, and then calcium activation, and then so there is a EET release, and all the whole works, right? These are the equations, about 89 equations. I hope even, biologists would be kind of moderately convinced by something like this. We did that. Now our main intention behind developing this model is to ask this question. Okay, forget about computation in the vessels. Can we even kind of strongly argue, this itself is a taboo right now. Can we have a signal going from vessel back to the neuron? It's always, in popular literature, right? It's neuron talking to the vessel. It's like a master-slave relationship. We talk about neurovascular coupling, right? Not vascular-neural coupling, right? So why is it like that? Why is it so one-sided? Because neurons are more dependent on vessels because they have to depend for the sustenance, right? So what happens to the feedback, right? Is the feedback informational? Is it significant? So we did this detailed model, and this is the firing activity of the neuron. There's lots of spikes, so it looks kind of all smudged. And then this is the pump currents. Then these are the calcium and IP3 levels in the astrocyte, EET release, then this is vessel radius, then glucose and lactate in astrocyte, glucose and lactate inside the neuron, then ATP fluctuation. So, okay, kind of you have everything going on right from, and the loop is closed. Okay, whenever the engineer says a loop closed, then you can't say which is source, which is destination, everything is going circulating, right? Okay, so we wanted to do a very simple study. Block the forward pass, right? And now only assume signal is going from vessel to the neuron. And to make it interesting, we assume that vessel has some oscillation. There's vasomotion. So only thing is it's a very slow oscillation. Okay, look at once in 5 seconds, it kind of just oscillates once. Okay, so if you do that in the neural activity, you see that little bursting emerging. So basically, without this, suppose there is a constant delivery of glucose, it will be a flat and regular phase. It's very simple. It's alternate type of neuron. But because of this bursting release of glucose, right, that is showing up on the neural activity. So now, okay, you can say that, okay, your food is coming in bursts, so I'm also active in bursts, and that's fine. That's one way of looking at it. But I can say that, I can I can maybe talk to the vessel, I can inject information to a vessel, and then listen to its effect in the neuron, right? Where do you draw the line and say, vessel is only carrying fluids and neuron is doing information processing, and it's very hard, which it gets harder and harder to draw that, can make that kind of distinction. So this is a detailed model of neuron-astrocyte-vessel interaction. One thing is vasomotion is shown to have a reverse influence. Okay, so now what do biologists say about this? Or is it just a kind of a hallucination of a model? Let us look at that. There's a very interesting idea called hemoneural hypothesis, proposed by Chris Moore, who was at MIT, and then he moved to Brown. I met him once, and because last I heard, like until 1 week ago, we were the only two guys who are talking about information going from blood vessel to the neuron. Okay, the general thing is information going from neuron to vessel. Neurons say, give commands, and vessels obey. So this guy wrote a review where he kind of garners evidence to support the idea of a signal going from vessel to the neuron. So, so whole bunch of things, and so through the nitric oxide released from vessels can also excite neurons, right? Because neurons also release nitric oxide, which can act on neighboring neurons, also accelerate plasticity and things like that. There could be mechanical engagement, so local changes in blood pressure can activate mechanosensitive ion channels. Right? So he gives possible mechanisms by which a signal can go from vessel to neuron. But here is a paper last week, which I appeared in Journal of Neuroscience, where the paper talks about vascular neural coupling. Okay, that's exactly what I was saying, right? So pressure increase in the vessel, right, in the arteriole, increased vascular tone, and simultaneously decreased the pyramidal neural firing activity, and vice versa. Okay, but here is talking about a signal that's going in the form of pressure getting transduced into electrical spiking activity. But in our model, it's through glucose and through energy fluctuations in energy delivery that is modulating neural activity. Okay, so we have a bunch of results, but all theoretical, and some experimental evidence from various labs, right? So what do we do next? Okay, so we still a very simple model. It's a very small system because you just have 3 cells, or 3 units, a single neuron, single astrocyte, single vessel. We need to scale up to a network before you can make some interesting statements. But then with 89 equations for one triplet, right, it's very hard to construct networks. So we said, no, we need to simplify this and did some simplification. So we said, okay, neuron is like a fast subsystem, and this is a slower system. Even glial oscillants are much slower than neural spiking, and vessel oscillants are even slower. So we just bundle this whole thing into a slow subsystem and express the whole thing into, in terms of a handful of not 89, but like 3 or 4 differential equations. And we did sort of a progressive reduction and simplification of the model, and so we published that. So with about 2 variables, you can get a reasonable this thing. So basically, under energy-starved conditions, you have like a nice bursting-like pattern of neural firing, and which is also supported by evidence. So we have that result. Okay, so I won't. Okay, I think I'm running short of time. So next we said, okay, so this I just want to push these ideas theoretically as much as possible, right? Because I don't know who will be interested in doing an experiment. So we propose another net model here. So one thing is that's well known is, so you have, so cerebral neurons, okay, talk to certain vascular regulatory neural systems. So there are deep subcortical systems, like for example dopamine cells, can also act directly on blood vessels and dilating them. Okay, and so we consider this kind of a network where neurons talk to this deep regulatory neural systems, which dilate vessel, and vessels talk to the neurons. So neurons can directly act on the vessels. This is local, okay, in a given, say, brain area, and vessels also by feedback act on neurons. So this kind of a system, we try to give it an interesting informational function. So in this network, this is an autoencoder network. So there's a given input X, and you try to reconstruct X. Y is supposed to look like X, and then the hidden layer now talks to nearby microvascular bed. So now this well-known work by Hinton and colleagues where they show that dropout gives you better learning of representations. So we use the feedback coming from the vessels to control dropout, and that is inspired by the previous work done by Karishma. This is a simplified neuron model, where we have shown that at low energy levels, that your threshold of firing goes up. That means that all things being said, neuron fails to fire if the energy supply is low, which is intuitively quite reasonable. So the feedback coming from the vessel oscillators, this is a network of oscillators. Vessels are modeled as a network of oscillators, as we have done before, a lot of other previous modeling work. So the output, you have an error signal. So that is, you have deconstructed output and the actual input, so you get an error. and we say that there is this little bunch of neurons which code for this error. That's kind of like your dopamine neurons, because we know from other literature, like from Schultz and all that dopamine cells code for some kind of a prediction error. So we use that idea here, and this controls the vessel oscillators, and that's also known from Iodakala's reviews on, right, dopamine cells controlling blood vessels. This is from the Nature review by Iodakala. So we put all these ideas together in a theoretical model, and shown that, okay, I don't have all the results here because really short of time. But what we have found is that if the activity here is desynchronized, right, you get rich representations in the input, in the hidden stage, hidden layer here. The activity is more regular, all synchronized firing, you get representations are not that good. Okay, this is quantified, you can look at the paper. So what is interesting in this is, you have a loop going on here, network output, then error computation, then vessels, and then neurons again. In the loop, I'm just putting vessels and neurons almost interchangeably. This is just a bunch of information processors. I'm not distinguishing at all. There's a bunch of units, right? And vessels just nicely come in part of this loop somewhere. One of the blocks in this loop of block diagrams. Now, is that kind of a picture valid? Right? Generally, vessels are always in the background supplying tea and coffee, right, to the guys in the foreground who are doing all the computations. But I mean, can we really put them as a part of these loops? I mean, that's something to think about. Okay, I'll move forward. So the other studies which we have done is, so this is a model of astrocytes, but what about other glial cells? I mean, we always thought, for example, macroglia are doing scavenging work, not very interesting. But macroglia also can chase ATP gradients. Now, why are they capable of chasing ATP gradient? Because that's energy stuff, right? So what about oligodendrocytes? They wrap around axons and control the conduction velocity. So, and it's also known that the oligodendrocytes are part of plasticity by which you can control the conduction delays. Okay, so we did a model of that where we showed that the same STDP, short-term spectrum-dependent plasticity rule can be used to control the learning of conduction delays on an axon, right, which probably is mediated by an oligodendrocyte. Okay, that's one study, and we just trying to write it up. And also we did a model of the effect of astrocytic feedback to the neurons on the oligodendrocytes. Okay, now what kind of experiments can you do here? One simple experiment is you give a stimulus like an oriented bar, measure from arterioles in a nearby active neuron. Can you find tuned responses in a vessel? For example, Mruganka's group have found tuned responses in astrocytes. Can we go one step further and look for such responses in a vessel? Okay, and not only that, if you give say a flickering input or a frequency input, will you find a oscillating output in the vessel? And what's the relationship between input frequency, neural firing rates, and the vessel? Can we really come up with a computational model relating all these three, at least in the feed-forward pass? Okay, that is one thing. Now, what about plasticity? So, can we verify? Can we do kind of a Bliss and Lomo kind of LTP standard classical LTP paradigm with astrocytes and vessels? I mean, you can define the experiment like this. So, you activate the astrocyte, you have calcium activation, and then there will be a vessel which is in contact with it and will dilate. Now, you also, so in a pre and post kind of simulation paradigm, you release a little bit of vasodilator on the vessel at the same time with a little delay, right? You are activating pre, you're activating post, do it again and again, a bunch of times. And after that, you activate only pre, the vessel should dilate even more than what it was dilating originally. That's LTP, right? Happening on the astrocyte vessel system. Can we do that? Okay, one more experiment I had in mind, which is, the motions work on, motion sensitivity in primates. So where they intervene and activate a neuron. So the monkey is looking at a bunch of moving dots, random dots, and they activate a motion sensitive neuron, and then they show the monkey makes a wrong judgment about direction of motion of the dots, right? So now, can you activate a blood vessel in the neighborhood and make the monkey make a wrong decision? For that to happen, the signal should be going from vessel to the neuron, so in the reverse direction. So that would be a wonderful experiment to verify. Okay, so up to here, I'm talking about mostly the modeling work is all published work, and then this is some work we have done with Mruganka in collaboration. So this is a master's student, Anjani. So the idea here is like this, problem here is you have a bunch of vessels, let's say whose positions are known, bunch of neurons whose positions are known. Now, where would you optimally place astrocytes so that there is optimal, some sort of a wiring minimization takes place? Okay, so we did that code, Anjani did that code, this kind of program, but we couldn't find much experimental data to validate it. So we kind of dropped that line of work, and then we looked at single cell, single astrocyte. Now there are, there's work by Kuntz et al. They've shown how you can explain neural arbors, right, by applying wire minimization principle. So he looks at two kinds of wiring, the wiring cost and the path length cost. and some balance, some linear sum of the two. And this cost is minimized for real neurons, and by playing with this one parameter, the balancing factor, you can reconstruct lots of arbors of real neurons, right? So we said, okay, can we try the same thing for an astrocyte? Okay, is there a violation kind of a principle at work in astrocyte? So we took data from literature, from Rodriguez et al., did some image processing, took this data and applied the same algorithm what Kuhns have proposed for neurons. And it works, and for a balancing factor of about 0.5, you get a very good reconstruction. And how do you validate this? So there are several parameters. There's branch order distribution, then path length distribution, and then Shaw intersection analysis. For all three, you get a good match at about balancing factor of 0.5. And then we also proposed another algorithm, but this work was incomplete. The student finished and left, and so we couldn't finish this work. Okay, some reconstructions. Okay. So then we also did some work on multiple astrocytes. If you have multiple astrocytes, how do you reconstruct all of them and so on. So, but that's not complete. So then I think I'll just skip this. I think there's a poster out there. So we proposed two models of tripartite synapse. You have presynaptic neuron, postsynaptic neuron, and the input coming from the blood vessel, from the astrocyte on one side, right? So the input from the astrocyte, we have assumed glial transmission, which is again a kind of a debated issue. So then we show how the feedback coming from the astrocyte controls your LTP and LTD of the synapse. So we have two models here. We just trying to write out lots of details for the model. Okay, I think I'll pass that. I can share with this. Is there in the report? I'll skip this. Now, so finally, okay, I just want to leave with these four ideas. The main hypothesis or a conceptual framework we're proposing is that brain's computation is performed by this triune network consisting of neurons, glial cells, and blood vessels. Information is going in all directions. Okay, so you need to have a much expanded theory of computation in the brain, which involves also glial cells and vessels. Then there's elaborate plastic network for distribution of energy resources in the brain. This network is adaptive, therefore it is tuned to support the kind of frequently occurring patterns in the brain. Any deviation from that, I think, so the plasticity is there to to take care of this deviation, right, through fresh learning. And understanding these distribution strategies can give insights probably into designing more energy efficient neural hardware. That's where I come into kind of CCBR in some sense. The inefficiencies in this delivery seem to underlie several important brain disorders. For example, I also work on Parkinson's disease and modeling basal ganglia. So people always thought that this dopamine cells in PD die because of pesticides and because of about 30 genes are involved and lots of other strange esoteric factors, risk factors. But over the last maybe half a decade, there's been a perspective developed by several workers who suggest that the neurons of this dopamine neuron, particularly in substantia nigra, are under very high metabolic stress, even compared to their cousins, like the neurons of ventral tegmental area, which are also dopamine neurons. SNC neurons are under much higher metabolic stress, and therefore vulnerable to cell death, for programmed cell death. I intuitively like this idea very much, and there is also similar work, suggestions have been made in case of other neurodegenerative diseases like Alzheimer's. Okay, I think I just suggest this, and I think I'll stop here. Can we come up with a whole class of neural models which combine neurons, astrocytes, and blood vessels, right? Not only at a small, a single column or a local level, but large scale level. That means, for on the vascular side, you need to also add your major arteries like carotids and vesicular arteries and all that, and their architecture. And now you ask the question, okay, now brain is doing some task and then something goes wrong, like vessel, macrovessel rupture, or some change in this, this whole interaction between vessels and neurons, right? Now, how do things degrade? So that means, so for example, look at stroke, which is a vascular problem, right? We always talk about what happens after stroke, but what has led to the precipitation of the stroke, right? What kind of a altered interaction between neurons and vessels has led to this precipitation of stroke? So can we understand that from dynamical systems point of view? Okay, so it brings up lots of questions, but can we think of a line of modeling, right? Also investigation, experimental investigation, which puts both neurons and vessels on a sort of equal footing, and that can also give rise to, I don't know about engineering applications of that, but that can probably lead to some insights into a better, more efficient design of neural hardware. Thank you.
Bring a few papers to your attention. So when I was at Bell Labs, in 1990, this is 20 years ago, kind of the very first data analysis I did with the fMRI data, I noticed these, what I call recognized as vasomotor oscillations, which you were pointing to, which at the same time were labeled in a different paper as resting state fMRI oscillations. So my paper got a lot of difficulty getting published because I was suggesting that the vessels are oscillating by themselves, as you know, they oscillate all over the body. And this has left me permanently skeptical of the resting state fMRI work, because you essentially have an oscillating blood vessel here, you have an oscillating blood vessel here. If you have two clocks, they'll synchronize. And so this has seemed to me a highly neglected area in functional imaging studies, actually treating the blood vessels, as you point out, as an active system. So I tried this 20 years ago, and during those 20 years, at every meeting I've been to where I can, I have raised some objections about the blood vessels being thought of as passive networks. I'm disinvited to those meetings, so people, but this is an important thing. So David Kleinfeld did some studies at that time, I can forward you those papers, where he looked at individual capillaries, the barrel cortex, and single viscous taps producing
They were trying to model that.
dilations, even at the level of individual
Columns.
blood corpuscles, one can see the neural activity, and more importantly, one can see the decoupling between the neural activity and the blood and the vascular thing, because the vasomotion, it is doing it by itself. Sometimes it couples to the neurons, sometimes it doesn't.
So that's what puts the whole fMRI data into question, because unless we get these interactions right, we can't assume that the vessel activity is a
I
mirror
can tell
image
you
of
that
neuron.
saying this has made me very unpopular. So there's a certain risk to pointing
huge
this
open
out,
question.
but it's an important,
Okay, thanks a lot. Thank you very much.